box, another, and of course the screen that you see on yet another monitor. I've got three monitors here. Okay, so we're doing nucleic acids and RNA. Let me test my pen, make certain it's working. And it is. Okay, great. All right, so the first thing I want to do is I want to talk about just the simple basics. What's DNA? What's RNA? What's the structure? So what's the difference between a pyrimidine and a purine? Now, pyrimidine is a single ring and a purine is a double ring. Let's take a look at that. So the pyrimidine has just the single ring, and the purine has two rings. I'm not going to get any deeper than that, other than you should be able to recognize these ring structures as one of the pyrimidines or one of the purines. So these are the base structures. So to take it to the next step up, and this is a problem that I still have myself. I keep tripping over the vocabulary. When you have the base only, we call it adenine, cytosine, guanine, thymine, and uracil. And chances are, as we go through the slides today, inevitably I'm going to mess this up myself. So it's I find it really hard to keep this straight. But as far as the pure naming convention, adenine, cytosine, guanine, thymine, and uracil are the names of these. So adenine, cytosine, guanine, thymine, those are the DNA bases. And in RNA, we use uracil instead of thymine. And of course, notice the only difference between thymine and uracil. That's it, the only difference. Once we attach those bases to the ribose ring, they become adenosine, cytidine, guanosine, thymidine, and uridine. I will tell you right now, I do not have any test questions on this because I myself mix this up. And if you catch me mixing this up, I will not take offense if you call me out on it, just so we're on the same page vocabulary wise. So I don't have any test questions on this because to me, this is splitting hairs. But as far as the pure naming convention, adenosine, cytidine, guanosine, thymidine, and uridine are the names we use for these when they're attached to the ribose ring versus when we're talking about just the base only. The reason we call it a base is because these are nitrogen structures, meaning they're very basic as far as pH is concerned. One other thing that I've gotten better, but the nucleosides are when we have the base attached to the ribose ring. But as soon as we attach a phosphate to it, it's a nucleotide. So tides have phosphate. So a nucleotide is a nucleoside with a phosphate attached to it. All right. Hi, I think I do have some of these questions in the easy generators, just so you don't trip over it yourself. And as I said, I find myself tripping over this all the time. All right, so what things do we need to pay attention to? Well, I said that the four DNA bases are A, G, T, and C. I'll save myself the embarrassment by using the one-letter codes here. A, G, T, and C. And the only difference between T and U is that methyl group. So thymidine has the methyl group, uridine does not. The other difference is the DNA bases have no oxygen on carbon number 2 of the ribose ring, where RNA does. All right, so once again, the base names, once we add the ribose, meaning the nucleosides, so these will be the nucleosides. And these are the nucleotides. Once we add the phosphate to it. All right, so now that we got that straight, and as I said, I'm certain that I'm going to mix these up as we go through this, and it's miserably difficult to keep this straight. Some of you are probably much better at it than I am, but these are the official names of these molecules. In addition to the DNA and the RNA, we find that these nucleotide bases are also found in the energy molecule of ATP. And the other interesting thing about ATP is it's not just a phosphate attached to it, but phosphoanhydrides, meaning a phosphate attached to another phosphate. Now, it's called an anhydride because we have to remove a water molecule to attach the oxygen of one phosphate to the phosphorus of the next phosphate by kicking out one of its oxygens to make that connection. All right, so what other molecules have these nucleotide bases? Well, the second messengers, which we talked about that in endocrine. We're going to talk about it again in neuro. We briefly discussed it in the BMS course at the very beginning. Cyclic ANP, coenzyme A, the energy molecule ATP, energy molecule FAD, flavin adenine dinucleotide, and NAD, nicotinamide, also known as niacinamide adenine dinucleotide. Now let's get into some really important stuff. One of the interesting things about DNA is not only do we have only 4 bases coding for the whole thing, we find that they form a double helix. There's your trick question for the test. What's the secondary structure of DNA? Answer, double helix. All right, if you miss that, kick yourself. So the secondary structure of DNA is a double helix. So that means it's a helical molecule, but it's 2 strands of DNA together. And they form hydrogen bonds, and that's what holds things together. What's interesting about these hydrogen bonds is the distance between them. When we have an A, it's going to attach to a T with 2 hydrogen bonds and a distance of approximately 11 angstroms. An angstrom is approximately the diameter of 1 atom. So that means the width of a DNA molecule is approximately 11 atoms wide. G is going to attach to C with 3 hydrogen bonds. And again, with a distance of about 11 angstrom. So the 2 hydrogen bonds and the 3 hydrogen bonds, that would be a testable question. How many bonds are between A and T? Answer is 2. How many hydrogen bonds are there between C and G? 3. So that means it's a tighter bond between a C and a G because of the 3 hydrogen bonds. What's even more interesting is we have enzymes that run up and down the length of the DNA checking this distance. Kind of like those trucks that run along the railroad tracks, checking the distance between the rails just to make certain that they're all the right width so we don't have a derailment. We have enzymes that run up and down the length of the DNA checking this distance to make certain we have the correct pairing. If we don't have the correct pairing, we're going to remove it. Now I have a whole section in today's lecture on how do we fix these mismatches. Another definition. These are all definitions, just so we're on the same page. When we synthesize DNA and RNA, we're going to attach the number 5 carbon onto the phosphate. I'm sorry. We're going to attach the number 5 carbon's phosphate is going to be attached to the oxygen that's on number 3 of the base in front of it. So therefore, the very, very first base has a phosphate that's not attached to anything. And the second base, its phosphate is attached to the number 3 position. So the convention is to say that we synthesize in the 5' to 3' direction, because on the very first end, the very first base, is a 5' phosphate that's not attached to anything. And the very last base has a 3' oxygen that's not attached to anything. Hence, we say it's synthesized in the 5' to 3' direction. And this is true for both DNA and RNA. So once again, secondary structure of DNA, double helix. All right, this side. I don't have any history questions on there. Watson and Crick won the Nobel Prize for the structure of DNA. They figured out it was not just simply a helix, but a double helix using molecular model kits. And they shared that with Maurice Wilkins, who was involved with the X-ray crystallography of this. The real tragedy of the story is the award was given in 1962, You have to be alive to win it. And the actual X-ray crystallographer was Rosalind Franklin, of course, working with all those X-rays, and probably didn't realize that we needed much more shielding. She died of cancer, I believe, 4 years prior to the award being given. So unfortunately, she never formally won the award. All right, so this is the famous photograph that was discovered if you take crystals of proteins or crystals of DNA or anything. If it's a helix, you get this classic X-shaped diffraction pattern. And from that hint, they were able to figure out it was a helix just because of the diffraction pattern they got from it. What was remarkable, it wasn't just a helix like an alpha helix in proteins, it was a double helix. Okay. All right, moving forward. In addition to the helix, we have additional higher structures called supercoiling, where the helix coils upon itself. This is going to become important when we talk about topoisomerase enzymes. So there's going to be positive and negative supercoiling. And this is because DNA is wrapped around protein. So eukaryotic DNA, anyhow. Prokaryotic DNA and mitochondrial DNA don't do this, but our nuclear DNA is so long it has to be tightly coiled, but those coils would create knots with each other, just like throwing a bunch of wires in a box. So we organize it by wrapping it around proteins called histones. And there's about 200 base pairs per nucleosome. So it wraps around each histone with about 146 bases, and then whatever, I can't remember the number now, 54 is it? Bases in the linker region, but a total of about 200 between here. Oops, I didn't draw that very well. Between here and there is about 200 bases. We call it the bead on the string model. There are 9 histones per nucleosome. One linker holding everything together, and then 2 copies each of the other 4. You do not need to memorize the names of these. Just know that they're basic proteins. That means they're going to be positively charged. DNA has that negatively charged phosphate backbone. They naturally are going to be sticking to each other. So notice the histones have a really high concentration of the basic amino acids, lysine and arginine. Really, really high. That makes these very positively charged. DNA is negatively charged, so it's just natural. It's going to wrap around these histones and stick very tightly. Then we coil it even tighter yet, and eventually it's getting thicker and thicker until we can actually see it in the microscope, and that is the chromosome, is a tightly wrapped molecule of DNA with all those histone proteins and other linker proteins holding things together. Okay. Next vocabulary, centromeres and telomeres. Centromere is the center portion of each linear chromosome and the attachment point when we double it. So right before cell division, each chromosome is going to double into an X-shaped looking molecule, not to be confused with the actual X chromosome, which has nothing to do with its actual shape. X and Y chromosomes do not have an X or Y shape to them. So that's a fallacy that I myself got caught up in. X and Y is just simply a designation. In birds, the gender chromosomes are W and Z. Has nothing to do with the shape. So X and Y chromosomes have nothing to do with their shape. But when we duplicate DNA, it goes from a linear molecule to a X shape where 2 strands of DNA are linked to each other by the centromere. Interesting, they're rich in A's and T's. Then we have protective caps, and that would be a test question. What's the name of the protective caps at the end of each chromosome? Those are called telomeres. That's the first one. That was all vocabulary. So just to make certain we're speaking the same language, that's all vocabulary. Let's move now into DNA replication. Now, where are we going with this eventually? Well, you're going to be clinicians. So we're really more interested when things go wrong. So right now we're talking about when things go right. What are they called? How is it supposed to work? And then we're going to eventually segue into what happens when it doesn't work right. And that's where you come in, helping to try to fix these things that don't go right. During the cell division or the cell life cycle, we'll start with what's called the G0. G0 means we got a cell, it's growing, and as it's growing, what we have going on is it doesn't need to replicate any DNA. But what happens when we either have a growing organism or there was cell damage and we need to replace some old dead cells? What we're going to do is we're going to go into what's called the G1 phase. The G1 phase is where we express proteins necessary for DNA synthesis. Then we have a restriction point. At that restriction point, we check the DNA to make certain that all of the DNA is healthy. We need to check to make certain there's no damaged DNA, no chemical mutations, no missing bases, no mismatched bases. That has to be all checked before we do DNA synthesis. Otherwise, we'd have mitotic catastrophe. If we get past the restriction point, we go into the synthesis phase or the S phase, and we double the chromosomes. So we have 46 chromosomes and they're linear. Then we go into the S phase, and at the end of the S phase, we have 46 pairs, or 92, connected at the centromere until we go into cell division. G2, we have now finished DNA duplication. We're going to prepare for cell division. And cell division takes place in the M phase for mitosis. And the single cell separates into two new cells. And that X-shaped chromosome, the centromeres dissolve and they pull apart, go to the poles of each cell. And now we have 46 chromosomes again in each cell. If we're done, we go back into the G0. If we still don't have cell-to-cell contact, we do another cycle. As soon as we have cell-to-cell contact, when the cells are touching each other, they stop dividing. That is one thing that goes wrong in cancer. We got cell-to-cell contact, but they continue to divide anyhow. That's called a tumor. Terminally differentiated is when we become so sophisticated that we cannot divide the cells anymore. Brain cells, heart cells, skeletal muscle cells go into the TD phase. They can no longer divide. And if they die, they're gone. All right. Now, when we do this division, we do what's called semi-conservative replication. And I'm hoping that all of you are reasonably familiar with this. A lot of this stuff I learned in high school. I learned it again in biology classes in college. So this hopefully should be relatively familiar to you, but I want to make certain that we're all on the same page. So if you know this already, great. I'm going to help you reinforce it by saying it again. If you're not quite remembering all these terminology or these names, that's why I'm going through it now, just to make certain we're on the same page. The parent DNA is a double helix. When we duplicate that DNA, we're going to unzipper it and match up the complementary bases to each side to create two daughter strands. So the new DNA for each new cell is going to be half of the old DNA and half newly synthesized DNA, hence the term semi-conservative replication. More vocabulary. I know the vocabulary gets tedious, but we need to speak the same language. Where we begin DNA replication, now here we're using a circular DNA. Circular DNA is going to be found in our mitochondria. Circular DNA is also found in prokaryotic bacteria. Now, even though we might say, well, we don't have we're not made of prokaryotic bacteria, that's not entirely true. Our gut and our skin are crawling with prokaryotic bacteria. hopefully most of it is helping us rather than hurting us. So the origin is called the ORI-C site, the origin site. That's where we have a lot of A and T pairs. A's and T's only have 2 hydrogen bonds. C's and G's have 3 hydrogen bonds. It's much easier to separate an A from a T than a C from a G. So therefore, the origin site or the ORI-C site is easier to separate because it's mostly A's and T's. Once we have it separated, we then do DNA replication times 4. We're going to have 2 replication forks going from the origin site. And in each direction, we're creating 2 brand new strands on the separated DNA. So 4 syntheses of DNA takes place at the same time, starting at each origin site. So 2 replication forks. At each replication fork, we have a cluster of proteins called the replisome. So the replication forks are right there. And the cluster of proteins at the replication fork is called the replisome. And those include proteins that do the DNA synthesis, separates the DNA, and does the DNA synthesis. When we continue all the way around in both directions, we eventually hit the termination site. If for any reason one replication fork hits the termination site before the other, it just simply stops and waits for the other one to catch up. And then we merge the 2 and we separate the DNA. In our nucleus, we have linear DNAs, not circular. And they're much, much, much larger. So therefore, we have multiple origin sites. And then they keep going in both directions. So we have 4 syntheses of DNA. We have 1, 2, 3, 4. So for each origin site, we have 4 strands of DNA being synthesized. We have 2 replication forks from each, so starting in each origin, we go in opposite directions from each origin site. So we have 2 replication forks, 1 and 2. And each replication fork is creating 2 strands of DNA. So a total of 4 strands of DNA is being synthesized at any one time. And then we keep going until we bump into each other, and then we eventually separate the DNA into 2 brand new strands. All right, that pretty much finishes up the pure science. Now we're going to start getting into application of this. Topoisomerases are a very, very important enzyme because wrapping and unwrapping, coiling and uncoiling the DNA is very difficult to do. So what it does instead is it literally breaks the DNA and reattaches the DNA to bring it through a strand, to kind of unknot the DNA, to uncoil it. Type 1 topoisomerases break just one strand and kind of twist it around itself. What's in this picture is a type 2 DNA where we break both strands, pull it through, and reconnect it. And that changes what's called the linking number, the amount of coils that we find in DNA. So when we talk about cancer, and that's where you're going to be going with this next, if we want to slow down cell division, we can block the DNA polymerase enzymes that are involved in those replisomes, but we can also prevent the DNA from unwrapping and separating. So that's why we have topoisomerase inhibitors, which are commonly used to treat things like cancer. They can also be used to deliberately slow down other rapidly dividing cells. So they could be used to treat inflammation. They could be used as antibiotics if they are specific for prokaryotes. And of course, eukaryotic topoisomerases will be mostly used as anti-cancer agents. Prevent the DNA from unwrapping. If you can't unwrap, you can't replicate. All right, so here's the first clinical thing I want to mention. Here's the name of 4 drugs. I will not ask you these drug names. but the pharmacologists may. So if these names come up again in your pharmacology lectures, you do need to know them. These are a list of 4 topoisomerase inhibitor drugs that prevent the DNA uncoiling prior to DNA replication. So we can't uncoil it, we can't replicate it. Okay. For example, here's one that's used with 5-fluorouracil. Okay, and it's used to treat small cell lung cancer. Can also be used with cisplatin. So they're generally used in conjunction with some of these other anti-metabolite drugs. Okay. This one is a topoisomerase 1 inhibitor. This one blocks both topoisomerase 1 and 2. And this one, I don't have the specifics here. I actually cheated and pulled these off Wikipedia, so you can look these up yourself if you like. I do not plan to test you specifically on these drug names, but I do believe you will have to know these drug names for both your other classes, especially pharmacology. All right, let's get back to that replication fork. There are 2 of them from each origin site. So one moving to the right, as we can see here, and a mirror image of another one moving to the left. At each replication fork, we're going to have the starting site, and then we're going to move in the 5' to 3' direction. We call that the leading strand. We got a problem on the lagging strand. We can only synthesize in the 5' to 3'. I said that about 20 slides ago. We can only synthesize in the 5' to 3'. So what we have to do is we have to skip about 1,000 base pairs. Lay down our origin and synthesize in the 5' to 3' direction. Then jump ahead about 2,000 base pairs and come back again to fill in the gap. Those are called Okazaki fragments. Now, that would be a potential test question. What's an Okazaki fragment? It is the fragments of DNA synthesis on the lagging strand. Why do you need to know this? Well, each one of these components of DNA replication is a site for blocking DNA replication. So anti-cancer agents are going to target these enzymes that are doing these processes. If we can block these enzymes, we can slow down rapidly dividing cells. That's exactly what we do with cancer. The downside with this approach is we slow down all the other rapidly dividing cells, like your gut lining, your skin cells, your red blood cell synthesis. So we talked about last week. For example, methotrexate is a anti-cancer drug that causes megaloblastic anemia. Okay, so that's going to be one of the problems with using these anti-metabolite drugs. So at the beginning of each molecule, we have what's called a primer. Again, why do we need to know this? Because each one of these is a potential target for cancer therapy. So a primer is how we start DNA replication. One of the really oddball things about DNA replication, we can't start from scratch. For some reason, we cannot lay down some DNA bases and start synthesis. So instead, we put down RNA bases instead. That's called the RNA primer. We go about 10 to 60 of them. And once we have that RNA primer in there, we can switch over to DNA and finish the synthesis in DNA. On the leading strand, no big deal. We put it down once and we just go for thousands of base pairs. On the lagging strand, we have to do this every 1,000 base pairs. So the first 10 to 60 bases in each Okazaki fragment has a primer. And what that means is we have to remove it. So this shows a replication fork. This one's going the other direction. And we have the Okazaki fragment. So at the beginning of the original leading strand and the beginning of each Okazaki fragment, we have a primer. That just simply means we have to remove it before we finish the job. So we have enzymes called polymerases. that do the original synthesis. The other thing about these polymerases, they act almost like a snowplow. As they're lay down bases behind them, they can knock out bases in front of them. Oh, I apologize for this coughing fit here. All right, now that's how both prokaryotic and eukaryotic synthesis works. What makes eukaryotic synthesis that much more interesting or difficult is in addition to all that, we have to remove all these histones before we do everything. So eukaryotic replication is pretty much the same logic as prokaryotic or mitochondrial, but it's just more complicated because we have a lot more proteins in there. They have to be gotten out of the way before we continue. And with that, I have now finished the first part, which is the basic science. Right. Any questions on that so far? Okay, now we're going to go into part two. Now, part two is DNA repair. All right. So actually, I cranked through this pretty quickly. Part one, I got through in 20, 30 minutes. Okay, so a question came in about the Okazaki fragments. Let me back up on that. So thank you for asking. The Okazaki fragments, it has to do with the quirk of DNA replication. We can only synthesize in the 5' to 3' direction. And everything's anti-parallel. So we are reading the template strand 3' to 5'. So this is how we're reading it. So we read the template strand 3' to 5' because we can only synthesize in the 5' to 3'. So the synthesis is 5' to 3'. On the leading strand, no problem. But on the lagging strand, we can only read in this direction. But we're going this direction. So we're essentially going upstream. We're like swimming upstream, trying to synthesize on the lagging strand. So since we can only synthesize in the 5' to 3' direction, and the lagging strand, and the template strand on the lagging side is in the opposite direction because everything's anti-parallel. And I failed to mention that before. This is anti-parallel. And I just spelled that wrong. Sorry about that. Anti-parallel. I think too many R's in there. Anyhow, apologize for my spelling. In the lagging strand, we cannot synthesize continuously. So we have to skip ahead and then come back in order to synthesize in the 3' to 5' direction, due to the fact that the other side, the lagging strand, is anti-parallel, meaning it's in the wrong direction. So we get around that by skipping about 1,000 bases, and then we synthesize in the direction that we can synthesize, 5' to 3'. But then we skip ahead from there about 2,000 base pairs, then back up to fill in the gap. Then skip ahead another 2,000 base pairs, back up 1,000 base pairs to synthesize. That is the concept of Okazaki fragments, where one side's continuous, the other side has to be synthesized in fragments. So it's a really bizarre quirk of nature. Chromosome number. I'm not sure what you mean by chromosome number. Yes, we are filling bases. Those gaps is DNA synthesis. So we synthesize about 1,000 bases. Then we jump ahead and come backwards to fill in the gap that was about 1,000 base pairs long. Fill in that gap. Jump ahead 2,000 base pairs, meaning there's another 1,000 base pair gap. Back up to fill in that gap. So we have to do these in fragments. Now, again, why would you possibly need to know all this information? Because the enzymes involved in doing these are all targets for chemotherapy. All right. So I'm not sure what you mean by chromosome number. Chromosome or synthesis direction is 5' to 3'. That you would definitely need to know. All right. So let's now move on to DNA repair. When we're doing replication, we unzipper and we have single-stranded DNA, and we find the complementary base. Wherever there's an A, we put in a T. Wherever there's a G, we put in a C. And we keep doing that, and that gets us the right distance. That distance should be about 11 angstroms, about 11 atoms wide. How does the cell know which side is the parent side? How does the cell know which side is the daughter side? Well, it turns out, and this is mostly between genes and not inside the genes. So in the non-coding region of the DNA, we have a lot of what's called methyl adenosines. It's just simply an adenosine with an extra methyl group. It does not affect the genes itself because this is generally in non-coding areas. It is just a marker, and it only occurs maybe once every 1,000 bases or so. And we have an enzyme that looks for this sequence, GATC. And when those four bases show up in exactly that order, it adds a methyl group to the adenosine. When we do DNA replication, the old side has those methyl groups on it. Now, this is not every adenosine. This is maybe once every 1,000 or so bases would have this, but just enough that we can recognize the difference between the old side and the new side. And after we're done, we'll then come back in and put the methyl groups on the new side so we can't tell the difference. But immediately following DNA replication, we are able to tell which side is old, which side is new. That's really, really important. If we do DNA replication and we get the wrong match together, say we put an A with a G instead of an A with a T, that can happen, and that does happen, but the distance is not right. The repair enzymes recognize the wrong distance, and it kicks out the wrong base. But how does it know which side to kick out? It kicks out the new side, the side without those methyl groups. So that's why we need to know which side is the old side, which side is the new side, because if we have a mismatch, we have to kick out the wrong base. So we need to know which side was new, which side was old, because the wrong base is going to be on the new side. So if we have a blip in the DNA, it is not the right distance because we have a mismatch. There are enzymes called methyl mismatch repair enzymes that recognize this and bind to the mismatch, the wrong distance between bases. It then curls up the DNA until it finds one of those methyl adenosines and nicks the new side, puts a break in it. And once it puts a break in it, it then chews up the DNA until we get to the bad base. Then we bring in the polymerases again, and I'm not going to bother talking about which is which. We bring in the polymerases and fill in the gap, hopefully doing it correctly. Now, it can happen on either side. It's going to do this to the nearest methyl adenosine, which again is only maybe once every 1,000 bases. So that's called methyl mismatch repair. Right? Now, it can be expensive. If the closest methyl adenosine is 1,000 base pairs away, we throw out 1,000 bases. But again, that's only one Okazaki fragment to fill in that gap and just to make certain that the DNA is held with high fidelity. This must take place at the end of the S phase. As soon as we finish doubling the DNA, we have to proofread it and look for all these mismatches and clean that up immediately. If we don't do it immediately, eventually those enzymes that add methyl groups will add them to the new side as well, and we won't be able to tell which side is new, which side is old. Now we got a problem. We got a mutation that was able to sneak past our defenses, and that's how the mutation got into place. So let's pause there. I want you to know what methyl mismatch repair is. Methyl mismatch repair is the method by which the cell can tell when we have a wrong matchup. If we have anything other than A and T or G and C, it's the wrong distance. We find it, identify it, remove it, and replace it. And it's all got to be done immediately after DNA synthesis. So think of that as proofreading. You're a proofreader. You're one of those DNA methylase mismatch repair enzymes. You're proofreading the DNA after replication to make certain that we don't have mutations. Now, of course, we know mutations do take place. So a few of these will always sneak through. But we want to make certain that this sneaking through is very much the exception to the rule and not the rule. All right. Here's a list of prokaryotic enzymes involved in methyl mismatch repair. And this is highly conserved. E. coli have all these methyl mismatch repair enzymes. Us humans have the human equivalents to these methyl mismatch repairs. And of course, it's much easier to study them in bacteria, but us humans have methyl mismatch repair enzymes. So we have a bunch of genes called MMR genes, methyl mismatch repair. And these are the gene names. If those genes are defective, we've lost our defense against mutations. And mutations are going to start to be much more frequent. And if you have more frequent mutations, your risk of cancer is much, much higher. So if you have a patient who has a gene defect in one of these genes, they are now vulnerable to developing cancer later in life. This is off the NIH site, okay, and National Institutes of the National Institutes of Health, the Cancer Institute site, describing methyl mismatch repair defects in humans. So the methyl mismatch repair deficiencies, these are most associated with things such as breast, prostate, bladder, thyroid cancers. And there's another one which we call it Lynch syndrome. I'm not going to go into that or test you on it. There are also gastrointestinal cancers seem to be more frequent. So what this does, it doesn't cause cancer. It just means we're not fixing the mistakes. So the statistical probability of developing cancer later on is much higher. Colorectal cancer is probably one of the biggest concerns for these errors. So what's on the test? What's methyl mismatch repair? That's the proofreading that takes place after each DNA replication. What happens if one of those enzymes involved in methyl mismatch repair is defective? We have a much higher risk of cancer. All right, so once again, methyl mismatch repair must take place at the end of the S phase, because once we get beyond that, we methylate the new side of the DNA and we can't tell the difference between the old and the new. And if that does take place, now a mutation has slipped through. All the other mutations we're going to talk about need to be fixed before S phase. That's called the restriction point. The restriction point means we cannot move on to the next phase until everything from the previous phase is fixed. And I do believe there is a restriction point at the end of S phase that prevents us from moving forward until we've fixed all our methyl mismatch repairs. All right, so here's just a list. You don't have to memorize these. Here's just a list of things that we know that cause chemical mutations. For example, there are sodium nitrite, sodium nitrate. These are commonly found in processed foods, deli meats. They're preservatives. Unfortunately, they tend to be converted into nitrosamines when they come in contact with stomach acid. Nitrosamines are notorious for inducing deaminations. Right? For example, it can turn a cytosine into uracil, or an adenine into a hypoxanthine, or a guanine into a xanthine. These are generally easy to detect because they're not one of the standard bases. There's another methylation that occurs mostly in the regions of the DNA that are in what we call the promoter region. And we see methylated cytosines in that region. And again, it's not all of them, it's just occasional. And it's for control reasons. But if that one were to mutate, now we got a problem. It'll get past our detectors because it's one of the standard bases. So let's back up. What do I want you to know? What is deamination? We've lost a nitrogen on one of our bases, mutating it into something else. What are alkylations? Well, adding a group where it doesn't belong. Now, we do add methyl groups onto our bases occasionally, but if we do it chemically, it's random. When it's done by our own enzymes, it's very strategic as to where it's found. But if it's put on there randomly, it can cause all kinds of problems. Depurination, we've just simply lost the purine. So let's cut to the chase here. So methyl mismatch repair, that has to be done after DNA replication to proofread the matches to make certain there's no mismatches. Base excision repair is what we're talking about now. This is to look for chemical mutations. So in other words, we've converted the base into something that should not be there at all. It's not just simply the wrong base, it's an abnormal base. So base excision repair corrects chemical mutations. That can take place at any time in the cell cycle. And here's a paper discussing how people who have defects of their base excision repair enzymes can have high risk of cancers. So base excision repair, again, it's found all the way from bacteria to humans. So very, very important. And defects are associated with neurological disorders and cancer. This particular paper talks about how people with this defect have a higher risk of colon cancer. Now, the right is colon cancer showing up. The lining of your gut has a high turnover rate. We're always knocking cells off of our gut lining. So we're always replacing our gut cells. So we have a high synthesis rate. And if they become mutated, they can become cancerous. All right, so in base excision repair, we're looking for a chemically mutated base. Unlike base excision repair, we don't need to know which side's old and which side's new. A bad base is a bad base, and we just recognize the side that has the bad base on it. We remove it, break the phosphate backbone. That's why there's several enzymes involved in each of these processes. We then chew up the adjacent bases, make a space in there, bring in a polymerase, and fill in the gap. In general, I'm just going to cut to the chase. That's called base excision repair for fixing chemical mutations. The next one, so we talk about mismatch repair, base excision repair. Now we're going to move on to one more called nucleotide excision repair. This is pretty much the same as base excision repair, it's just for larger mutations, most notably pyrimidine dimers. So here's one, is talking about people who have a defect of nucleotide excision repair. And here's a name I would like you to know. Xeroderma pigmentosum. That is one of the most notorious of the nucleotide excision repair defects. And most of these dimers are caused by exposure to sun. And these dimers are actually caused by sunlight. These patients are highly at risk of skin cancer. And sometimes they're referred to as children of the night. They cannot go outside and expose their skin to sunlight, so they can only go outside at night. So here's one disease name I will ask you to put into your vocabulary note. Xeroderma pigmentosum is one of the highest profile disorders that is a defect of nucleotide excision repair. And these patients have an extreme sensitivity to sunlight because they cannot fix UV-induced damage to their DNA. So what is this UV damage I'm talking about? Well, if we have two thymines, okay, adjacent thymines, there's a chemical reaction that causes a cyclobutane ring to form between two adjacent double bonds. So if we have two bases next to each other and a photon of light hits this just right with the right UV wavelength, it can cause this dimerization. It's like fusing two of your bases together. It puts this gigantic kink in the side of your DNA. That would be catastrophic if the DNA was to go into replication. That's got to get fixed. So we have a set of enzymes called the nucleotide excision repairs that look for these giant kinks in the DNA. These are found all the way from bacteria to humans. It's that important. It looks for these lesions. It cuts out a section of DNA, larger in humans, of course, where the defect is found, brings in the polymerases, and replaces that section of the gene, hopefully with, not just gene, but the DNA, fixes that section of the DNA with brand new DNA to hopefully repair the damage that was done. If you have a defect like xeroderma pigmentosum, you cannot do this. All right, so you will need to know the difference between mismatch repair and when to use it. That's for right after DNA replication is proofreading. Base excision repair to correct chemical mutations. Nucleotide excision repair to correct bigger mutations, usually ultraviolet light-induced lesions. There's a fourth type called direct repair. This is limited. We don't get to use this one as much, but it makes the most sense. It just simply reverses the errors that were created. So it's just an alternative to the other methods. For example, there's a way to undimerize those thymines. And as long as those enzymes are working good, we can do corrections without having to replace all that DNA. So I'm just going to cut to the chase and say that's known as direct repair. There's another one that takes those alkyl groups, like those methyl groups, off the bases that are not supposed to have them. Now, some do have to have them because it's a signaling method, but if it's just randomly put on there, we can remove them. All right. Number 5, crossover repair. What if these other repair methods just don't work? Something's wrong. We do have one more method to fix things. This is for what's called strand breaks. What if we have an uncorrected repair and we're doing DNA replication and then we get stuck and we stop because we can't go beyond the repaired site or the damaged site? It then breaks the replication and we get what's called a strand break. So the newly synthesized DNA is missing DNA. That is potentially catastrophic. That, of course, could kill a cell. But we have a way to get around that. It's called recombinational repair. So this is the fifth and last one I'm going to talk about. So methyl mismatch repair is the first one. Base excision is the second one for chemical mutations. Third one is nucleotide excision repair. That's for like ultraviolet induced mutations. There's direct repair, which is not used as much, but it's an elegant way of just reversing the other errors. This is the fifth one. This is for really big ones. We have to concede defeat. We have lost a chromosome. A chromosome has been destroyed. But we get our chromosomes in pairs of 2. So in recombinational repair, what we do is we borrow DNA from your other chromosome. You get one from your mother, one from your father. If one is, say the one you got from your mother is damaged beyond repair, we can copy the DNA from your father's side. And of course, it can go the other way around as well. We can copy DNA from the other parent's side to fix the damaged chromosome. That's called recombinational repair. There are some disorders where those enzymes are defective and they have a really high profile name. The BRCA1 and BRCA2 errors. BRCA1 and BRCA2, these are errors that prevent you from doing this recombinational repair. BRCA1 is probably the most high profile of it, and most associated with high risk of breast cancer in both men and women. Obviously, it's going to be much more of a concern in women. It's going to be much more common in women, but it can happen in both men and women if you have this BRCA1 error. It is so because it creates such a high vulnerability to cancer. It's not uncommon for people who have been diagnosed with this BRCA1 error. So the BRCA1 gene is working fine, you're good. But if the BRCA1 gene is defective, there's a very high probability of developing breast cancer over your lifetime. Right? And preemptive surgery has been common in people who have this BRCA error. But it's also associated with cervical, esophagus, liver, stomach, and uterine cancer. BRCA2, which is a parallel disorder, also meaning that the crossover repair is defective, is highly correlated with high risk of pancreas, prostate, and melanomas of the skin. So I would expect you to know that what's a BRCA error? A BRCA error is a defect of crossover repair. All right, so let me pause here. 5 types of repair systems. Know what each one of the 5 types is used for, and know that if they're defective, of course, not with genetic screening, 23andMe, Ancestry DNA, all those things can detect if you have these errors. And if you do, you now have an elevated risk because a defense mechanism is missing. All right, that's the second part. All right, I'm cruising along. I've got through essentially 2 hours with a lecture in an hour's time. So we could take a break, but I think I'm close enough to being finished where we can just simply slog through this and finish early. Any questions on this so far? Yes, you had to go home and make sense out of this, but is it organized well enough for you where you can just simply say 1, 2, 3, 4, 5? These are the 5 things I need to know. And if any one of those goes bad, you're at higher risk for cancer. Okay. All right. Now we're going to move on to P53 errors. And I have to believe that you've heard P53 multiple times already in other lectures. P53 is most associated with this restriction point. That's the one that stops the cell from going into S phase until the DNA damage is repaired. And it does so by affecting molecules called cyclins. Cyclins are proteins necessary to permit the cell to go from one phase of the cycle to the next phase of the cycle. If P53 is elevated, it blocks the one that goes into S phase. Specifically, it's called cyclin E-CDK2. So P53 blocks cyclin E-CDK2, which is the cyclin needed to allow the cell to enter S phase. I'm going to spare you the headache and just cut to the chase here. P53 is also known as a tumor suppressor protein, meaning if it's functioning properly, it suppresses tumors. If it's defective, you've lost that protection. So if there's DNA damage, and we have all those enzymes going up and down the DNA, checking to see if it's the right distance, to make certain all the bases are the correct bases. And if it finds an error, it triggers an increase in concentration of P53. P53 triggers a cascade that's going to inactivate that CDK2 cyclin E. If the P53, I'm sorry, if there is no error, P53 does not activate, and it allows passage from G1 to the S phase. Okay. You can go through the rest of this to kind of make sense out of all that, but I just want to cut to the chase and say, if we have DNA damage, P53 goes up and we inactivate the complexes. If DNA is not damaged, it's good. We don't elevate P53, and we're able to pass from G1 to S. Under normal circumstances, if the P53 goes up, it blocks movement into S phase, and it stays there until the damage is fixed. But what if the damage is beyond repair? The cell just simply cannot fix its DNA. If we stay in that phase long enough, it triggers apoptosis, which is programmed cell death. So if we cannot fix the DNA, the cell is programmed to kill itself. That's apoptosis. As opposed to necrosis, which is physical damage to a cell. The cell has been crushed, burned, chemically damaged. The components of the cell are damaged. That's necrosis. Lack of oxygen, lack of blood, that's necrosis. But we cannot fix the DNA. The cell will automatically kill itself, so it does not become cancerous. That's called apoptosis. If that goes wrong, of course, elevated risk of cancer. This slide, I don't really have much on in terms of the test. It may complement some of your other teachers, but necrosis and apoptosis, necrosis is just damaged cells. Apoptosis is a normal cell with bad DNA, just simply killing itself off, so it doesn't do damage to the whole organism. So let's go back to this tumor suppressor gene, P53. If it's damaged, you're vulnerable. And it's the usual characters here. These are all known to elevate in people with P53 mutations. Because P53 prevents the formation of tumors. But if we have damage to our DNA and P53 is also damaged, the damage does not get corrected. And the cell does not go into apoptosis. Now the cell can become cancerous. All right. That pretty much finishes up the discussion of the 5 types of DNA repair concerns, the role of P53. And just as a side note, what if we do have one of these errors and we don't correct it, and we get a mismatch that gets past our repair mechanisms? Well, for example, here's a repair that did not get corrected. Say we have a C to G, and it's supposed to stay C to G, but we have an error that does not get corrected. Now we've flipped it. So now we have got a mutation that got put into our DNA because we were unable to correct an error. And that's how mutations take place. They should be rare, but we know they do happen. All right, let's finish off. I've got 13 more slides to go, and we're done for today. What's the Ames test? Ames test is a test for mutations. This is a little bit difficult to get your head around at first. Mutations are actually quite common. But here's a bacteria that's been grown to be unable to make its own histidine. Now, our cells cannot make histidine because all humans are histidine defectives, meaning we have to have histidine as an essential amino acid. But bacteria can make all 20 of their amino acids. And here's a bacteria that cannot make histidine, and it can grow on histidine-rich plates. But what if we put it on a histidine-deficient plate, a bacteria that cannot make its own histidine? Something bizarre happens. If the mutation is just simply a single point mutation, one base, random back mutations can take place. It can unmutate. So there might be thousands, maybe millions of bacteria on this plate, and occasionally one will back mutate, and each of these spots represents a colony of cells grown from a single cell that back mutated and now can make its own histidine. It was just a single point mutation. What we're now going to do is we're going to add to this plate a new chemical. You're testing a new drug. And we put this new drug on a piece of filter paper right in the middle of the plate. If we see an increased number of mutations showing up at a very defined distance away, meaning at just the right concentration, that implies that your new drug causes mutations. That also means your drug is probably not coming to market. It's dangerous. If we lower the dose and the ring comes in, lower the dose, the ring comes in closer, I'll tell you right now, your drug is not going to market. It potentially causes cancer. Why? Because there's a 90% correlation between mutagens and cancer. Does that mean some mutagens don't cause cancer? Yeah, but at the same time, you still don't want mutagens because mutagens can also cause birth defects. All right. This is out of your textbook, so I'm going to just gloss past this one. All right, we're almost done. Now we're back to vocabulary. What's the difference between an allele, a polymorphism, and a mutation? An allele is variations of the same gene. So, for example, I've got an allele that gives me brown eyes, and you've got an allele that gives you blue eyes, okay? They're just different alleles. I think eye color is a little bit more complicated than that, but it was just an easy, easy example. Variations to the same gene in the same individual. If you remember, I mentioned that we have multiple copies of the same gene. We get 2 chromosomes, one from our mother, one from our father. We could have one allele from our father and a different allele from our mother. That just means there's a few bases in that DNA sequence that are different. Polymorphism. More than 1% of the population has that same allele that you have. So that could be eye color, skin color. It's just a polymorphism. It represents more than 1% of the population. But if it's a really rare allele, it's less than 1% of the population, we call it a mutation. We're really bad about these definitions because mutations get used much more frequently than 1%. But technically, anything that's more common than 1% is called a polymorphism, which is just simply considered a variation in the population. A mutation means a rare difference. Now, not all mutations are bad. Some mutations might be good. If you got a mutation that turns you into an Olympic athlete, well, not necessarily a bad mutation, is it? Okay. Single nucleotide polymorphisms. The single nucleotide polymorphism is a single change in the DNA. One base has changed. And this is old news. We talked about this last week when we did the hemoglobin. When we have a single point, we call it a point mutation. Some of those point mutations may not change anything. It could be in the non-coding region. of the DNA. No change. It could be inside an intron. Now, who cares? We cut those out and throw them away. But if it's in the exon, the part that gets coded into protein, but it doesn't change the amino acid because we know there's redundancy in that codon chart, all of those are silent mutations. By the way, this will be on the test. So know the difference between a silent mutation and a missense mutation. We're going to subdivide missense into conservative, non-conservative, and these are gray definitions. Conservative means a big change. Conservative means small change. I got that back. Small change. Somebody emailed me over the weekend regarding this one. They had it backwards. Now I just made the same mistake talking about it. Conservative is a small change. Non-conservative is a big change. Think of these definitions as bookends with a lot of gray in between. Okay? So if it's a small change, it's conservative mutation. If it's a big change, it's non-conservative. The best example is sickle cell anemia, converting a negatively charged polar glutamate into a non-polar uncharged valine. That's a non-conservative mutation, makes a big change. Conservative mutation means something that would convert maybe like an aspartic acid into a glutamic acid. Those two amino acids have the same chemical properties. It's just they differ by one methyl group. Then there's the nonsense mutations. Okay. Just as a side note, those hemoglobinopathies that we talked about, those were all missense mutations. We've changed an amino acid. The thalassemias we talked about, those were nonsense mutations. We completely messed up the protein. It's all the wrong bases. Either the protein's too short, early termination mutation, or it's too long, it's a run-on mutation, or it's really messed up. The frame shift got off because we either added a base or removed a base. So those are the nonsense mutations: early termination, run-on mutation, and then the frame shift mutation, which could be due to either an insertion or a deletion. All right, now let's go into some stuff we didn't talk about last time. Duplication mutations. This means that sometimes a gene inside your DNA gets duplicated. And this has happened numerous times throughout history, and sometimes we just have redundant copies of the same gene. Other times, too many copies of the same gene can cause problems, such as trisomy 21, where you got one too many copies of the chromosome 21. Now you're overexpressing proteins, and it causes all kinds of problems. So a duplication mutation could cause overexpression of certain proteins, which can cause problems. A repeat expansion. There's a section of DNA where a bunch of bases just repeat themselves, and they repeat themselves maybe 20 times. Then it goes up to 100 times. That's called a repeat expansion. That could completely disrupt the function of a protein. So here's two more definitions. Two other things that can go wrong. We can duplicate genes, or we can expand the size of a gene by putting a bunch of repetitive DNA in the middle of it. All right. This I can gloss through because this is simply a repeat from last week. And just to refresh your memory, this was a little bit nicer. I did a better job on this one versus the previous PowerPoints. This is an example of silent mutations. Every single thing inside these black boxes represents silent mutations. Okay. Some get more than others. Okay. So there's nothing special about the color code other than just a visual way of keeping track of which ones are which. So inside these boxes, notice that leucine has 6 different possibilities. Notice that methionine only gets 1 possibility. So leucine gets 6. Methionine only gets 1. Isoleucine gets 3 codes. So there's redundancy in the code. So if you make a change that doesn't change the amino acid, silent mutation. If we have a missense mutation, we've changed one to the other. Conservative means we're going from like glutamate to aspartate or leucine to isoleucine. Non-conservative means we're completely changing the value, the polarity and the charge. Of course, the classic example is sickle cell going from glutamate to valine. And glutamate to valine, I believe, is this one here. That's the normal code, and that's the abnormal code. Okay. Early termination, we stick a stop code on where it doesn't belong. Run a mutation, we turn a stop codon into a different amino acid. Frame shift means we've lost our reading frame because we've added or deleted a base. Of course, the frame shift makes a real mess because now we got all the wrong amino acids from that point forward. And that's it. 93 slides in less than an hour and 20 minutes. Boy, I was flying here. Good thing this was recorded. Now, I don't really do a lot of the active learning stuff that some of the other people do. I pretty much do that in the easy generators. And I've got a request from some of your classmates that they preferred me just doing a summary style review of this. And so that's what I did today. Okay. All right. Any questions on that long list of things to know? So going back to this, part one was what's the structure of DNA? So in other words, what's it supposed to look like? What's it supposed to be called? And I've got about 5 questions on what DNA is supposed to look like. Histones, centromeres, so on forth. Because I've only got 5 questions to cover a lot of material, I do have several matching questions on the test. So the beauty of that is partial credit. And the other beauty of it is I can ask more than one thing within a question as opposed to just a random grab of a question. Okay. DNA replication. The big thing about DNA replication is how do things work? And then, actually, I take it back. The replication was considered part one. So part one is on the 5 questions on DNA replication and just the structure of DNA. Part 2 is DNA repair. So those are the 5 ways in which we fix errors. And what's more relevant to you and what's going to show up on the test is what happens when those repair mechanisms go wrong. So we know those 5 different ways that things can go wrong, and there's 5 questions. So imagine I could try to have a question on each of the different type of repair mechanisms. In addition to that is the P53. P53 is what stops the cell cycle so these repair mechanisms can take place. Okay, and part 3 was on P53, kind of filling in the gap, the P53, the Ames test, and then just repeating these mutations. You know these mutations are going to be on there. I covered it twice, so that's definitely going to be on there. So yeah, 15 questions from the 93 slides. That's exactly right. All right, so question is, if you study from the slides and the easy generators, that should put you in pretty good shape. I'm going to say that if you do all my practice questions in the homework, all my practice questions on the easy generator, you should at least I would hope, pass the test. But I really hope that you aspire to do more than just pass. I'm hoping that you really want to ace this thing, at least ace my section on it. So I would suggest that what you do is, in addition to being able to answer all the questions, look at all the in-between stuff. What other questions did I not ask that I could have asked? And if you have a study group, maybe you can give each other additional practice questions, try to second guess what could I have asked that I didn't already ask in those practice questions. So I think the practice questions should get you at least a passing grade. At least that's my hope. But if you want to ace it, you need to fill in the gaps as well. All right. Most importantly, understand it. If all you do is memorize it, it's gone by tomorrow. We all know that. But if you are going to look at the logic, what's the logic of how these things work? That's what you remember. And that's what I remember over the years. Now, of course, the recording I think would be good study material. I think the questions are your best study material because it really goes right to the point of what I'm asking you to know. So my questions I would say would be the most powerful focus on what you need to know. Make certain that you can answer those questions, but don't just know the answer to the questions. That'll do you in. You want to know why the answers are what they are. So if I word it differently, which you know that I will, you have no problem. You still understand the question. So even if it's worded differently, it shouldn't faze you whatsoever. So if you all you do is memorize the answers to questions, you'll get into trouble. Especially if I reverse the logic or twist things around. But if you understand why the answers are what they are, it should make no difference how I ask the question. You, as long as you understand it, you answer the question. Um, all right. So yes. So 15 questions from this last set. And then the other ones, I separated them. There's four of them. So four PowerPoints, 5 questions each. So 20 points from the other four PowerPoints. So 5 each for the other four PowerPoints, 15 from this one because I combined 3 all into one big giant PowerPoint, parts 1, 2, and 3. And that's it. 35 questions are already submitted. And hopefully, it's my wish that all of you smoke the doors off my section of the test. And if you can smoke the doors off the rest of the test, that's even better yet. All right. I'm looking at the chat box. So since the videos take the longest to go through, I would go through the videos on the Easy Generator first because it's got those questions interspersed within them. If you haven't already done that, I suggest you do that. The practice questions that I posted, I'd like you to try your best to stick to the due dates for at least doing them for the first time. But of course, you can go back and repeat them. Even if it marks you late, that's just a quirk of the program. I'm not going to call anything late until after the exam. So you've got up until the time you start the exam to do those practice questions multiple times. Okay. And I just got a reminder to put up a post for the research. I will give you a link to the faculty research page. I'll have to be honest with you, I got to go find it. It's hidden somewhere in my Google Drive. And I'll just give you it's a public link. I'll just have to give you a copy of the link so you can go in there and take a look at it and see what's currently going on. And if you're interested in research, contact one of the teachers that's doing research. And there is a fall and spring elective for P3 students in the DE program. We currently have 3 DE students in my current program. We got 6 people in Florida, 3 in DE, and 3 in Erie. So I got 12 students in the current research course. It's a wonderful number. It's a perfect number. If you'd like to be in this small class for next fall or spring, you're welcome. If you just want to do research for the sake of research, that's also a possibility. All right. So once again, I'd say there's a question on here. What's the minimum you need to do? Hopefully you do more than just the minimum. Do the practice questions. I suggest you do the easy generators with the practice questions. Then go back and do the questions again in the homeworks, because I've duplicated a lot of those questions in the homeworks. And you get the immediate feedback on that, of course. And that's how we go. Somebody asked, do I publish often? And unfortunately, I don't really publish that often because my primary focus is teaching. Okay. All right. Anybody else have any questions? And I'm impressed with all the questions about research. That's really nice. But let's get back to what we're doing here today. And that's the DNA stuff. I think Dr. Kearns is next with cancer. So she's going to take it to the next level up. So hopefully all those vocabulary helps you with her lectures. Any last questions before I end this session? All right. We're finished a little bit early, and I look forward to seeing some fantastic grades on the test I think it's next week, right? I'm looking forward to seeing some fantastic results on the test next week. All right. Have a great day, everybody, and I'll be looking forward to